Dear students, let us study Bryophyta classification. Let's do the classification of Bryophyta as proposed by G.M. Smith. Division Bryophyta is divided by Smith into three classes, and these classes include Hepataceae, Anthocerati, and Muscae. Hepataceae he further divided into orders. He included four orders in it: Sphaerocarpales, Marcantiales, Jungermannials, and Calobriales. Whereas in Anthocerati, he included only one order, that is Anthoceratids. In Muscae, he did not directly group it into orders, whereas he divided it into three subclasses. And these subclasses included Sphagnobraea, and Andreobrea, and Eubrea. Sphagnobraea he further grouped into one group only, the order Sphagnales. In Andreobrea he included only one order, Andreales, and in Eubrea he included Eubreales. Nowadays they group it into Polytrichiales and Phenarials. He grouped it only into one group, one order, that is Eubreales. Now let us study the characteristic features of Hepataceae. It is derived from a Latin word hepatica, means liver, thus the Hepataceae are popularly known as liverworts. The vegetative thallus, that is gametophyte, is usually dorsiventral and is either a thallus, thallose, or a leafy axis, foliose. When foliose, the leaves are without midrib, rhizoids without septa. Rhizoids are present but they are without septa. Each cell in the thallus contains many chloroplasts. The chloroplasts are without pyrenoid. Anatomically, the gametophyte is either simple or composed of many tissues. A photosynthetic tissue on the dorsal surface of gametophyte is always present, and the cells forming it have numerous small chloroplasts without pyrenoids. Sex organs are embedded on the dorsal surface. Sex organs usually form from a single superficial cell. Sporophyte may be simple, as you have seen in case of Riccia, having only a capsule, or differentiated into foot and capsule, for example, Marcantia, Palea, and Porella, etc. Capsule lacks columella. The spores on germination usually directly or in some cases indirectly give rise to the gametophytic thalli. Now let us study the simple type Riccia from Hepataceae. See, the thallus looks like a rosette. It is dorsoventrally flattened, growing on the substratum prostrate. The dorsal surface is dichotomously branching and provided midrib, darker in color as compared to the ventral surface, which is lighter in color, and provided with rhizoids, both smooth-walled as well as tuberculated, and rhizoid and scales. The vertical section shows the photosynthetic region, upper one, and the lower part, which is somewhat parenchymatous, the assimilatory region, provided with lower epidermis, and the photosynthetic region provided by upper epidermis, which is not continuous, provided with air pores and air canals. The cells of the photosynthetic region contain chloroplast and without pyrenoid. Now, the male and female sex organs, the antheridia and the archegonia, are borne on the dorsal surface. They are rather embedded in the dorsal surface. Here I am showing you the archegonium. Similar way, the antheridium is also borne on the dorsal surface. And antheridia produced antherozoids, which are biflagellate, containing the male gamete, and the archegonium is a flask-shaped structure with a neck and the bottom. It's called venter. The neck is provided with a lid cell, a lid which comprises of 2 to 4 lid cells. The venter contain venter canal cell and an egg, and neck is provided with neck canal cells. When the egg matures, The venter canal cell degenerates, sorry, not venter canal cell degenerates and neck canal cell degenerate, producing a lot of mucus, which puts pressure onto the lid cells, and lid cells open, they're pushed apart, and the lots of mucus also comes out, and and also an opening is there through which the male gamete makes an entry. It's actually the biflagellate anthrozoid which casts off its flagella and just moves into the neck, and the movement is facilitated by means of the mucus which is formed as a result of degeneration of the neck canal cells and the ventral canal cell, and the male gamete ultimately reaches the bottom of the venter and fuses with the female gamete, producing a zygote. Zygote is the first cell of sporophytic generation. After some time, the neck also degenerates and a sporogonium or a sporophyte is formed, which is actually the venter. Zygote is diploid. It is the first cell of sporophytic generation. It only multiplies by mitosis, giving rise to many cells, and the outermost covering is called now calyptra, and a simple sporogonium or a sporophyte is formed in Rixia. The nuclei so formed are formed as a result of mitosis, so all of them are diploid. Each is called a spore mother cell. Each spore mother cell undergoes meiosis, forming a tetrad first and then spores. Each spore so formed as a result of meiosis is haploid. When the spores are mature, lot many spores are formed inside the sporophyte. The calyptra ruptures and the spores are liberated out. They fall onto the substratum, the moist soil, germinate and give rise to the gametophyte. We'll stop here, continue in the second video. Thank you to all of you for patiently listening. We'll continue the hepatitis in next video. Thank you. Please like it, subscribe it and share it. Thank you.